Respected Deshpande sir, our Chief Advisor of All India Drugs Control Officers Confederation, Dr. V. Raju, Vice President Regulatory Affairs, Wajan Pharmaceuticals LLC, New Jersey, USA. Our today's guest speaker, Lakshmi, Coordinator of All India Drugs Control Officers Confederation Training Academy, Central Council members, senior regulators, friends from the pharma company, and scholars and students, distinguished participants, ladies and gentlemen. In this virtual webinar, I recognize some familiar names of participants. It's my sincere privilege and welcome to you to this important webinar, Pharmaceutical Quality System. As we know, science and technology, manufacturing and production continue to evolve at a record pace. We also need to adapt emerging technologies and latest guidelines to strengthen our inspection approaches and assessment so that we can stay ahead of the industries we regulate. Pharmaceutical quality system, QS, are designed to provide high quality finished pharmaceutical products with QA and PQS working together in synergy. A robust PQS is critical to assuring drug products are manufactured to meet desired quality and performance attributes. For a pharmaceutical product, typically we cannot see the quality. Quality is inbuilt and it travels towards infinity. So what do we expect from quality? The ultimate aim is to satisfy the end user, patients and caregivers assume their drugs, which are safe, efficacious, have the correct identity, and the deliver of such performances as described in the label, and also perform consistently over the shelf life. I hope this webinar would be a great opportunity in enhancing mutual understanding in the conceptual and also theoretical aspects of pharmaceutical quality systems. Together, let us accelerate the exchange of ideas and scaling up of good practices. Let us take this opportunity to hear from Dr. V. Raju, who is expert on this subject and having vast understanding. Actually, Dr. V. Raju was a very familiar presence of Indian regulatory system attached to AP drug department, very vast experience in auditing pharmaceutical companies, CGMP compliance, document management, training curriculum development, evaluation, and execution. Dr. Raju was very active in All India Drugs Control Officers Confederation activities and was editor of All India Ad Hoc newsletter and very knowledgeable person in US FDA matter. With these few words, I, on behalf of All India Drugs Control Officers Confederation, sincerely welcome Dr. V. Raju sir. Kindly give your valuable deliberation and insight into the subject. Thank you all.
Thank you so much, Dr. Jayanta sir, for the opening remarks. Now I take the pleasure of inviting Dr. V. Raju sir, as pointed out by Dr. Jayanta sir. Dr. V. Raju sir has been associated with not only with the drug control department but with the Ad Hoc Academy, Ad Hoc activities since the inception in 1995. And he has a vast experience of almost 38 years, both in the drug control department for about 18 years and about 20 years in the pharmaceutical industry on the other side of the table, and especially in various positions right from the product design and approval and life cycle management. So during his tenure in the industry, he has filed and got approvals, almost couple of dozens of ANDAs and DMFs of different kind of pharmaceutical dosage forms. And he has very successful track record of completing many US FDA audits without any 483s. And he has traveled across the globe for the vendor development and CMOs and CROs. So this is brief about the professional activities on the pharmaceutical industry sector. but though he has been moved to USA, he is closely working with the Indian Pharmaceutical Association and ADCOG, and he has served in different roles for the development of pharmaceutical profession in the country. He was the active member, and he served as a general secretary of Indian Pharma Association of AP State Branch for almost 3 terms. And he was also the founding secretary and treasurer of IPA Regulatory Affairs Division for 3 terms. And with the ADCOG, he has associated with different levels, and he has been actively contributing to the Indian Pharmaceutical Congress hosted by the ADCOG at different occasions. And he served as a secretary for the LOC in 2000, and also served as a core committee member of the local organizing committee for 66 IPC held in Hyderabad, hosted by ADCOG. Sir, we are so glad to have you with us, sir, on this occasion of webinar on pharmaceutical quality system, and we look forward to hear a grateful insights from you on this subject. Pleasure to welcome you, sir.
Thank you, Lakshmi. Thank you, Jayanta. It's a very pleasure, always to associate with my alma mater. ADCOG is my alma mater. Whatever I learned as a member of the Drugs Control Administration here, working in the state as well as working along with the central officers, like stalwarts like Venkateshwarlu, the Krishna Dev. So the many stalwarts, we worked with them when the industry was in, budding stage. So it was, I was fortunate to work with, stalwarts like, Deshpande ji, and the founding members of ADCOG during the 95 IPC at Vizag. So I have fond memories with the ADCOG as well as the Indian pharma industry. I'm fortunate to see them again here, Deshpande ji, Jayanta, Bhaskar, Lakshmi Prasanna, and all other friends we have logged into today's webinar. It's a wonderful technological advancement, so we could see each other, but sitting at different corners of the country or the world. So it's always a pleasure, speaking on the platform of ADCOG. I always love it. So whatever I learned, I try to share my experiences, as part of my learning process in India as well as in the USA. So let's begin. And if you have any questions, so please, follow the procedure. Like I think type in the questions, and Lakshmi Prasanna will read out, and if time permits, we'll answer all the questions. Thank you, and let us start the presentation. Today, we're going to talk about the quality systems. So it's nothing new to all of you because we are part of the system that is regulated here. So when it is regulated, so you are bound to play in a design space. The design space could be quality or specification, whatever it is, whatever name you call it. So it is a system we are already inside. So when you are inside, so we have to play by the rule book. The rule book could be the Drugs and Cosmetics Act or the Drugs and Cosmetic Rules. That is the primary governing law of the land. So the quality system, so we are all they're familiar, but I want to refresh the memories, refresh the knowledge we have, how we can better the lives of the patients who consume our products. So this is a famous saying by a French playwright in the 17th century. So this applies aptly to every field. It's not only for our regulators or the manufacturers. You are held responsible for what you do, but also for what you do not do. So this is a big paradigm. You'll be questioned if you did not do what you're supposed to do, but you'll also be questioned for not doing that, why you did leave that. So this is a big dilemma for everybody. So we have to be careful when you are playing in the field of pharmaceuticals. We'll be questioned on both sides. So you have to have a balanced approach on this. So what is quality? So this is again a perception. Each one defines his own quality. Last time, I think a couple of years ago, I met one of the top manufacturers here who got a warning letter, import alert from US FDA. So I happened to talk to them, and they were worried, why they got it. And one of the points they mentioned was, they questioned me, what is the relationship of a pressure gauge with the quality of the API produced? That is a question asked to me by the CEO. I think FDA cited the non-functioning or non-calibration of the pressure gauges on the vessels. So they asked me, my product complies with all the specifications. In addition to the pharmaceutical specification, they have their in-house extra specification. They say that my product meets all or exceeds the specifications. Even then I was cited for a malfunctioning pressure gauge. So that was the question put to me. Then I asked him again, so what do you think, what do you feel is quality? The quality is something you check at, or I put it in a colloquial common words. Some people buy product like a shirt I buy from a particular XYZ store. I feel if I buy from that XYZ store, I feel that it is quality. But if somebody else will feel that if he pays XYZ amount in a particular store, he feels it's quality. Means if he pays, say, 5,000 rupees for a shirt, he thinks it's a quality for him. But for me, if I buy at a particular store, I feel it is a quality. So quality is the perception. So ultimately, either it can be anything of the four I cited, but the last one is the one suitable for the patient. Whatever patient thinks is quality is the quality that we have to accept. Fitness for intended use, that is the quality. So how do you make a product fit for use? So that is the biggest challenge for all of us involved in the manufacturing, distribution, logistics of all the pharmaceuticals here. So fitness for intended use is the quality. That is the quality the patient wants, and that is the quality we have to accept. So can the patient see quality? He cannot see quality, only he expects quality. Nobody can see quality. You see, it's only an expectation. So how do you expect this? So why do they expect quality in this? Because they cannot ensure quality by themselves. They entrusted this responsibility to all of us, the regulators as well as the manufacturers, because we are all qualified, trained, and authorized. So authorized to do certain responsibilities, certain functions, authorized to make a good product. And we have sufficient training. We have a lot of qualifications. We studied all sorts of chemistry, pharmacy, pharmacology, biology, biopharmaceutics, pharmacokinetics, statistics, mathematics, whatnot. That comes into the playing field to make a decision in the pharmaceutical manufacturing. So the common man cannot ensure themselves about the quality of the product they consume. So they have delegated this responsibility to the regulators as well as the manufacturers. So prescribers and patients, they want the product to be safe and effective. So I don't have to define the safety and effectiveness. Safe means it doesn't cause any other effect other than what it is intended. Then effective means it is delivering its job. A delivery in the same performance as stated on the label. If the label states particular ingredients, those ingredients are only available on the product. Nothing else is present because some people could be allergic to some components of a product. If the listing on the label does not specify what all the ingredients, then it could be dangerous to some people. So this is exactly what it delivers as stated on the label. And it is made in a manner that ensures quality. And also it should be available when required. So these are all the assumptions the prescribers and patients on drugs. So all these put into a single concept, a quality product. So it's a the expectations and the assumptions of the prescribers and patients on a product is the totality of all these: safety, effectiveness, delivery, and quality and availability. This is all put together is a quality. So it's whether we are making the product by design or by accident or testing into quality. So these three important things we have to understand here. The quality is by design, or quality is by accident, quality is by testing. Whatever is feasible and whatever is practical, we should not adopt. Quality should be by design. It should not be tested at. Quality should be built into the product rather than testing at end. Testing is only assurance, but the quality is not built. Quality is built by design, and it should not be an accident. Accident should never happen. So some of the things what we see in the media and press, bothers us, whether the product is properly designed and at the end properly evaluated. So how do we ensure this? How do we build this quality in the product? By following certain design principles. It's quality by design. QBD is quality by design. So when you understand the design principles of anything, like the design space you make for a product, then you need to understand, so what is the product life cycle? This is the design space. The product life cycle, if you understand properly, then you will understand how the quality can be designed, how can the quality be built into the product. So what is the product life cycle? Its typical life cycle, looks like this. It's somebody thinks of introducing a product, like launch conception. So we want to launch a particular sustained action product, or a meter dose inhaler, or a nasal spray solution. So that is a concept. So when you have a conception, when you dream of making a product, then the first thing comes into mind is the selection of the availability and the sourcing of the active ingredient. So that is the active ingredient. So when you identify an active ingredient that is suitable to develop a dosage form, then you need to characterize the API. So what are the characterization techniques available? So what are the critical attributes in an API that matters a lot in the performance of the product? So that characterization helps us in understanding requirement of the product as well as the qualities of the API. So this we need to balance when selecting an API for a particular dosage form. So once you characterize, once you identify the attributes, then you make trials. That can be called as a pre-formulation or whatever it is. So you develop a concept, you test a hypothesis. So you formulate a product and see that. So this is what I'm looking at. This is, you derive certain attributes for the product. So that attributes are designed in the pre-formulation or not, we'll evaluate like a proof of concept experimentation here in the pre-formulation stage. So once the pre-formulation is okay, then the pre-formulation, it's like a tentative formulation. So tentative formulation is simultaneously used for analytical development. It is not sufficient if you develop a product, but it has to be tested with a suitable analytical method. So these two things in the industry generally goes hand in hand. Once the formula is locked in, that formula, the qualitatively and quantitatively, so the same formula is used for developing analytical methods. So once the analytical method goes on, then the product development goes up. The product development means it could be the pilot studies. It could be scale-up studies. You have to decide the size of the batch. It could be, say, 100,000, 1 million, 500,000 tablets or capsules, or 500 liters or 5,000 liters. So that is, you have to have a scalability technique. Your product developed should be capable of scalability. The scalability is achieved once you have a robust formulation development. So once the formulation development is completed, the analytical development is completed and validated, the scale-up batches are made. Scale-up batches could be anything. your commercial batch or 1/10 of the commercial batch. Anything is okay. The scale-up batch, you make it, you put them on stability studies. So to evaluate the shelf life, to understand the product performance during the shelf life, whether this could interact with your container closed system, or will the product be stable, exposed to the light, or exposed to higher temperatures, or exposed to humid conditions, or dry conditions. So all these, the points are evaluated during the stability studies. Then after the stability study, the application comes for approval. So they file with the regulators, the Drugs Control Administration or CDSU or any whatever it is. Once the application comes for approval, it follows a review process. A review could be anything, either digital review, electronic review, manual review, paper review, whatever it is. The review, then after that, during review, there will be some questions on the API characterization, API selection, API source, or the attributes of the API, or the process steps, the process parameters, how they are arrived at, the product attributes, what are the target product attributes the company is seeking. So all these questions, if the reviewers have questions, so they communicate to the applicant. So the applicant has to answer satisfactory to the reviewers. So all the questions regarding the API or the process or the excipients or the product and the stability and the shelf life. So the questions are answered, that is a compliance submitted. Then again, the compliance is reviewed. If the compliance is okay, it can be approved, or if there are further questions, they send back to, again, the review questions. The cycle goes back. The review cycle, the approval cycle stops. Then the questions are again sent back. Again, they seek that till it is satisfactorily answered. Simultaneously, when the review goes on, the field inspection also goes. Sometimes here, the same inspector does both review as well as the audit of the facility. Whether that particular facility, which the product is sought to be manufactured, is capable of manufacturing, whether do they have sufficient space to store the components of the product? Do they have sufficient space to store the packaging material? Do they have right storage conditions for API and the finished product? So this inspection determines the scalability as well as the feasibility of manufacturing at a site. So that is the compliance part. Compliance from the review, compliance from the field audit or the inspection. So both together, if both concur, then the product is approved. So the product is approved, then the commercial manufacture, then the distribution, and the last point is the continuous monitoring. This is the pharmacovigilance part. You get a lot of questions from the market, a lot of complaints, a lot of adverse event reports. So all these are monitored after the product is launched till the product is discontinued. Even if the product is discontinued for a period of at least 3-4 years, depending on the shelf life of the product, that needs to be monitored continuously. So this is a learning process. If there are certain complaints, certain different aspects of this, so that will come back to the manufacturer through the regulator, maybe. So they evaluate the effect of all these on the product life cycle. This will help in bettering the product acceptance in the market. So this is the typical product life cycle. So these are various stages. So whether we follow this or not, that depends on the individual companies and the thing. But this is the general industry followed product life cycles. Then, so what is the science behind product development? We need to understand a lot of things. Chemistry, so either organic chemistry, inorganic chemistry, physical chemistry. Why you need to know organic chemistry? Because the synthetic part of the drug molecule, how it could cleavage to produce different related substances or impurities. Those impurities could be toxic. Then the certain catalysts, like inorganic catalysts, if there are residues of these catalysts go into the product, what happens? Then physicochemical properties of the product. So the physical chemistry part is useful in understanding the spectral characteristics of the molecule. Then pharmaceutics, I don't have to emphasize because most of you have studied this. Pharmaceutics helps us in formulating the product, a better product. You understand the dynamics of the interaction between the excipients and the active ingredient, how the process affects the API. The chemical engineering part is in the process development, whether the developed process is scalable, reproducible, and robust. So this chemical engineering helps us either in the synthesis of a molecule or in the scaling of the drug product manufacturing. Then you need to understand toxicology, pharmacology of the molecules and their pharmacotherapeutics. So these are the scientific aspects of any product development and product selling in India or anywhere in the world. So this is the science behind a product development. We should understand all the intricacies of how they play in making an acceptable product. So product development starts with, as we mentioned, selection of API, then selection of excipients, the process aids like filtrates, or the inorganic gases, when you fill a liquid product, either it's injectable or so nitrogen or carbon dioxide sometimes, how the manufacturing process, and what is the packaging system for the drug delivery. If it is a drug device combinations like metered dose inhalers or dry powder inhalers, nasal sprays, there are many drug device combinations. The packaging system also plays a very robust role in the product development. So excipients, there are different grades of excipients. Lactose is available for many grades. MCC, microcrystalline cellulose, is available in different types of grades like particle size distribution, surface areas. All these, they matter a lot in terms of the drug delivery. Then the product development also involves, as we saw in the product life cycle, the analytical methods, their development and their validations, and the stability of the product. And the last point is the cleaning between the batches and between the products. We often see, so hundreds of products are permitted for a manufacturing company. So we are not sure how the cleaning is effective or not, whether they have a proper cleaning method or not. The cleaning between the batches of the same product or between the products, different types of products. So there should be an acceptance criteria. The previous residue of the product should not be carried beyond a certain limit, like 10 ppm, or this is an acceptable limit, 10 ppm, depending on the molecules you make in a particular facility. So the cleaning of the equipment, cleaning of all the filling, packaging, transfer equipment, then the cleaning agent used, like a detergent or the IPA, the residues of all these matter a lot in terms of causing a reaction in a patient. If the API is not properly cleaned from the previous batch, if it carried to the next product, it could cause a fatal reaction for somebody else. You don't know. If this is not ensured properly, it could be fatal for somebody else. So the cleaning is part of the product development. Cleaning means not the physical cleaning. You should have a proper cleaning method, and the method should be capable of removing all the residues like your detergent, the API, the chemical residue, and if you are using any sanitizing agents like alcohols. So the cleaning method should be capable of removing all these agents. So the cleaning method should be capable, and it should be so robust, should be able to detect up to picogram or nanogram level. Then you could detect the chemical residues. So API selection. Again, whether you go for a particular company, particular country, it all depends on your requirement for the product, how it's available, the source of supply. Because once you develop a product with a particular source and a particular grade of API, that should be continued throughout its shelf life or its life cycle. If you want to change this, then again, you have to go back to the drawing board. You need to redesign, you need to reevaluate the effect of the change of source on the product performance. That is a big thing. That's a very big thing. That's the reason when you select an API, you should always make yourself sure that the source is reliable and they could supply the material, whatever you want, the quantities, the quantity of the material you require, the tonnage of the API requirement. If they are able to meet that, you should select such an API source. Otherwise, you have to go back to the drawing board, redesign the product, again put it on stability, evaluate it, and then you launch it with a new source of API. So another important aspect of the API is the impurities or related substances. These are very, very, sometimes they're very toxic, sometimes they're carcinogens. Even the structure activity relationship, there are throughput analysis, it could predict whether it could degrade to a particular related substance which can be mutagenic or carcinogenic. There are certain functional groups identified. A lot of literature is available, libraries available to understand each chemical, how it can give rise to its degradation products and degradation products toxicity and mutagenicity or carcinogenicity. So the selection of API, you should be very careful of the impurity profile of the API. If the two sources are available with lesser number of impurities, I prefer to go with the lesser number of impurities containing API, even though it costs a bit more, because it saves your time in analytical method development and validations, and it saves a lot of questions during review. So the impurities are very, very critical, the quality attribute of an API before you select an API. Then the residual solvents. There are certain solvents which are prohibited, which are carcinogenic, like class 1, class 2, class 3, you guys are aware. So it's better the class 1 solvents are avoided. And class 2, class 3 solvents, yes, with certain limits, you're allowed to have these in the API. So you have, when you have more solvents are used in the manufacturing of the API, then more bothersome for the user, like the manufacturer of the drug product, because you need to identify the solvents, you need to develop a method which is suitable to detect those solvents and quantify those solvents. So again, this is also a critical factor in deciding the source of API. Then the key starting material. This is the one which contributes to the functional group of the API molecule. So key starting material is the major contributor for the molecule. So this needs to be ensured that it's properly manufactured by in-house or a third-party supply. So third-party supply also needs to be monitored about the manufacturing practices at their facility. And the last one is GMP. So it goes without saying. So the typical things to look into the API source is the impurities and the residual solvents. These are two important chemical attributes of the API before you select for making a product. Then the characterization. So how do you characterize your API? How do you decide which source of API is suitable for your intended product? So you want to make a inhaler, a meter dose inhaler, like a dry powder inhaler. So particle size is very critical, because that should not get deposited in the lungs. So particle size, even if the product is a tablet, extended release tablet or a delayed release tablet, particle size makes matter worse. So particle size is very critical in terms of the dissolution. If you are making a suspension, the dissolution makes a lot of difference if the particle size is not uniform. So particle size and size distribution. So particle size distribution, so how many particles of a particular size, D50, D10, D90, so whatever size, so you want. So that has to be decided based on your end product quality. So this particle size is an important characterization, you look for in the API. Then the solubility. Solubility of API makes a lot of difference in the bioavailability. If it is freely soluble, then the bioavailability is easier. If you want to make a solution dosage form, this makes your life easier. But if it is pH dependent, then again. So pKa is very important here. If it's a pH dependent solubility, then you need to look for the dissociation constant of the API. Then Another important point is the polymorphism and chirality. So these are the very, very important things when you're making a suspension dosage form or a oral dosage form with a wet granulation. So polymorph, chemically it's the same, but the structure, crystallinity changes. I'll tell you my own example. When I was doing my PhD, we were using a molecule. I wanted to see whether my formulation is comparable to a reference product. When we conducted pilot biostudy, once I developed the formulation, my product did not release anything. I did not see anything in the plasma. We spent a lot of money on this, but it did not result anything in the blood level. My product failed in blood level, but then we went back to the drawing board. We tried to identify what caused this problem. This is way back 25 years ago. So then we understood that particular molecule has two polymorphs, one polymorph 1 and polymorph 2. Polymorph 2 is insoluble. Polymorph 1 was soluble. So that unfortunately, what API I got, I did not characterize. So that I got was polymorph 2. I've used it in the formulation. I took it for the biostudy. It failed miserably. Then we identified that. Then we came back. We tried to procure the form 1. Then we reformulated. Then we reevaluated. Even in the formulation, we evaluated whether the 2 theta values match with that of the reference standard or not. Reference standard as well as the drug product which we are referring. So then when it matches, then we got the comparable blood levels. So polymorphism is very, very critical. So there are many examples in the literature. If you go and search in the Google, you find the effect of polymorphism on many products which were withdrawn from the market because of the polymorphic change in the suspensions and oral solution dosage forms or dosage forms, tablet dosage forms, which were manufactured using wet granulation techniques. So chirality, again, chirality is very, very important. You must have seen there are many molecules were approved, both racemic as well as chiral, cetirizine and levocetirizine. Cetirizine was a racemic and levocetirizine is chirally pure. Same loratadine, desloratadine. Nexium brand, that is omeprazole. So omeprazole, Nexium brand, esomeprazole, yes. So Nexium brand, omeprazole is Prilosec, Nexium was esomeprazole. So this chirally active molecules play a lot of role in the bioperformance of the product. So API characterization, these should be remembered, the polymorphism and chirality, the particle size while formulating. Then the stability of API, of course, it goes without saying. If the molecule is not stable, so you're not going to end up with your good expiration date for your product. If the API starts degrading, so here the stability we have to understand is how it degrades, under what circumstances, at what pH. It degrades in the acidic condition or the alkaline condition, or when exposed to light or when exposed to temperature or oxidation. So these are the establishing the robustness of the molecule, whether it withstands all these stress factors like acid, base, light, temperature. So the molecule has to withdraw without giving rise to degrade to related substances. So that stability of API is also very important for the formulation development. So this is an example, is from the literature. Ritonavir is a protease inhibitor for the HIV treatment. This was launched by, I think, Abbott in 1996. So Abbott launched this as a soft gel capsule with ethanol and water, solution of the molecule in ethanol and water. So during 98, I think there were a lot of market complaints that the product did not dissolve properly. It failed in dissolution. The Abbott identified by evaluating their retained samples and found that dissolution was not meeting the specification. Then again, so they understood, when they investigated, they understood that it turned into a different polymorph. So then they understood that there are two polymorphs for this ritonavir. Polymorph 1 they thought, but the polymorph 2 was 50% less soluble than it was thought. So the product was withdrawn from the market. There was recall. It cost a lot of money. It damages the reputation of the company. Then they reformulated into a stable oily vehicle so that there is no polymorphic conversion. So this is one example in the literature. Another example is about the atorvastatin. This is very commonly used molecule for reducing the cholesterols, Liptar brand. So this is about, in the literature, you see about 27 polymeric forms for this product. If this is not properly assessed before it is converted to a tablet, so tablet performance is questionable. So the marketed product is amorphous, it's not crystalline even. So that has to be decided before you convert this into a formulation. So if you do not understand these, so you're bound to get, some people complain that it's not working. So why it's not working? Maybe it's a different polymeric form. It doesn't release the drug it's supposed to release. It doesn't dissolve in the intestine. So to get a job. So atorvastatin is known to contain around 27 polymorphs that are reported in the literature. So solubility is another important character of the API you look into. So if poorly soluble APIs or result in poor dissolution rates. Poor dissolution rate affects your bioavailability and bioequivalence. Because the drug is not available in solution at the site of absorption, so then it affects its bioavailability. And these drugs are very prone for the food effect. If somebody takes it on after eating breakfast or lunch, then the poorly soluble drugs will be affected its bioavailability. Then certain formulations like injectables, solution injectables, or the oral solutions, or the topical solutions, it's difficult to formulate so that the stable solutions, monophasic solutions. And this analytical method development, again, as we saw in the product life cycle, goes hand in hand with the product development. While you characterize the API, while you do the pre-formulation studies, feasibility studies, proof of concept studies, and simultaneously the analytical method development and the validation. So any analytical method should be specific. That means it's able to identify that particular API present in the formulation. Then it should be precise, it should be accurate, and it should be robust. So there are other requirements, but these are the 4 important aspects of an analytical method. It could be UV method, it could be any method, HPLC method or LCMS method or whatnot. So whatever instrument you use, but the minimal attributes of a good analytical method, suitable analytical method, are it should be specific, precise, accurate, and robust. So if any of these do not meet, then the method needs to be tweaked. The instruments, what we use, they have to be qualified. They have to be periodically calibrated. And it should be capable of recording the metadata that goes into decision making than the audit trails. So any analytical instruments, what we use in making a GMP decision should be qualified. I think you know what is a qualified. Qualified means it's suitable to use for its intended purpose. That means it works as designed. It works as it was purchased for. So it should be qualified. It should be calibrated periodically because we have to see that the instrument performs the same way when we purchased. So these calibrations ensure that the machine performs in the desired way. And you should be capable of recording all that has gone into the system. When you put a sample for testing, you should record who has tested it, at what time, what date, and what are the values that recorded. Maybe area in a HPLC, the peak areas. Based on the peak areas, you derive the result, assay. So this metadata should be stored permanently. and it should be capable of audit trails. Audit trails are like footprints. So we should go back and see whether who has done this, when it was done, whether did they repeat anything without any reason, any logic, or any failures that were not reported, or any data which should have been taken out or not taken. So all these are stored in the audit trails. So we should be capable of accessing the audit trail when we see a result, when we question a result, when a result is reported, we should go back to the metadata on the audit trail and see whether everything is okay or not. So sometimes in the industry, we saw in many of the 483s that they are running a trial, a separate experiment, separate folder, they store the data. If they pass, then they run the sample in the GMP. So that lot of hundreds of examples available, hundreds of citations available in the internet about this missing data, not reporting the data, not recording the audit trials, the metadata is missing. So there are many questions, different types of audit observations, that you notice on the internet. If you search internet, you find hundreds and hundreds of citations on the missing audit trail information on the decisions we make. So method, the instrument, and now the challenges for dosage forms. As we talked, so these are very, very challenging to make suspension to emulsions. In the suspensions, they are thermodynamically unstable. So to make it stable, you use a lot of suspending agents, or you viscosity building, and then the particle size of the API is controlled, particle size distribution is controlled. The same thing about emulsions. Emulsions are like globules. So oil in water or water in oil, globules. Again, globule size distribution, globule size estimation. They matter because they form the sediment. When it forms a hard sediment, the uniformity of the dosage is a problem. So they are very, very challenging dosage forms. And if you determine that the particle size and particle size distribution, or globule size distribution are critical, then it should be evaluated periodically from time to time during the shelf life of the product. Then the sterile products, again, another challenging thing. The sterility assurance is the biggest challenge for pharmaceutical manufacturing. So all the practices you use, the people we employ are to be properly trained on the sterility and the sterile principles of the products, how a sterility could be breached. So this is a very challenging dosage form. Then extended release, modified release, sustained release products. These are again very, very challenging. And we believe sometimes, the way we make the products, ER becomes MR, MR becomes SR. So we don't know whether if the product is not properly designed, if the process is not properly evaluated, processly developed, then it could be a very challenging a dosage form. Then other one is the metadose inhalers, dry powder inhalers, nasal delivery, topical delivery. The topical patches is the biggest challenge again. And skin is the biggest barrier. It doesn't allow the drug molecule to penetrate easily. So it's a very, very challenging dosage form. Then the last but not the least, proteins, peptides, and biologicals, vaccines or monoclonal antibodies, whatnot. All these, they're very, very challenging dosage forms in terms of its characterization, in terms of reproducibility of manufacturing, in terms of robustness of the dosage form developed. So they are very, very challenging dosage form. They're not that simple. A lot of science goes into evaluation. So we should be equipped with a lot of information when developing these challenging dosage forms. So products attributes, these are the product attributes. We were discussing about the API. The product attributes are these are the 5 things. Quality by design. Quality, target, product profile. When you develop a formulation, you assume or you decide on certain quality attributes, targets. You want my product should contain, these are the target profiles. My product should dissolve not less than 80% in 15 minutes, or my product should dissolve in 15 minutes in 0.1 HCl, not more than 5%, and pH 6.8 buffer after 2 hours, not less than 90%. So that is a profile. You're putting a target for your product. So this could be anything. You have to decide before you develop a product. So this QTPP, quality target product profile, is what you're going to see for the product. Then CQS, so critical quality attributes. What are the attributes you want in this product? My product has to be free from residual solvents, or my product has to be free from impurities, or my product has to dissolve this much in this much time, or the product should release the drug at so-and-so interval. So these are the CQS. Then CMAs, this is what we were talking about, the excipients and other formulation aids, material attributes. So there are different types of excipients we use in developing a formulation. So each excipient plays an important role, and it could affect the stability of the product we use and the performance of the product. Certain excipients like sodium starch glycolate or CMC curve. So these excipients play a major role in the performance of the pharmaceutical. Then CPPs are the critical process parameters. So how the manufacturing process is achieved, how robust is that, how reproducible is the process. So if the process is not reproducible, you get a lot of variability in your product produced. Then the last one is the strategies, control strategies. How do you control your product performance? What are the specifications you want the product to meet? So these are the control strategies. So these control strategies are the final one and most important thing. So based on all the CTPP, QTPP, CQS, CMA, CPP, and the control strategies. Then the stability. So once you devise the product is evaluated. What are the storage conditions? You want to store the product in room temperature or refrigerator condition or freezer condition. It depends on your QTPP. So target profile. So the storage condition, expiration dating, how long this product is going to last on the shelf. It's 2 years, 18 months, or 36 months, depending on your QTPP and the evaluations. Sterility, there is no other though. It's either sterile or non-sterile. There is no semi-sterile here. So if a product is sterile, it should be sterile throughout its shelf life. Then the dose delivery, whether the dosage form is delivering what it is supposed to deliver, a particular time, a particular period of time. So if it is a extended release product, it should start a loading dose and the maintenance dose. That should deliver. If it is a nasal spray or a meter dose inhaler, every actuation should deliver, say, for example, 100 microgram of the API or 150 microgram of API. It should deliver plus or minus whatever is your C control strategy should deliver the same dose every time. So how do you ensure this? So this has to be evaluated in vitro. The actuation force. So there are different techniques available in evaluating nasal sprays or meter dose inhalers. Then what is the interaction of your product and the container closure? Is there any danger that is coming from the container closure? If this is true, especially for liquids dosage forms, the interaction between the container closure and the product. If it contains solvent like alcohol or something else, then it could leach from the container. If it is a polythene container, HDPE or PP or PET, some of the ingredients of your the container, plastic, could leach into the product. That could be dangerous, and that could destabilize the product. So we have to evaluate the interaction of this container closure with the product. So in the real-time situation, during the shelf life, if it is not sufficient, if you established at the time of release of the product, But if the product is labeled its expiry as 24 months, they should evaluate at the 24 months as well, 12 months in between, 24 months as well, the leachability or the extractable profile of the container closure. So that plays very important role in the stability as well as the safety of the product. Then some of the molecules are light sensitive. So we need to evaluate how stable, how the container closed system protects from the light. So your container closed system should be capable of protecting the product from the effect of light. So that is photostability. And thermal cycling, like freeze thaw, because the product moves from one temperature cycle zone to the other, like Jammu Kashmir, Himachal Pradesh, these Himalayan states, the temperature goes to close to minus below zero. So, but whereas in the south, it could be 40 degrees. So how the freezing and thawing. So freeze for some time, then thaw it, freeze for some time. Some cycles you have to determine, ICH has guidance on this. The product, especially the liquid products or semi-solid products, should be evaluated for this thermal cycling effect. Freeze thaw study. Then in-use study. The in-use study is one when the product is like, as I mentioned, it's a drug device combination, or it's a dry powder liquid oral. That means a dry syrup. So you have to reconstitute before using the product. So after reconstitution, how long this product is stable? So that has to be established. That is called the in-use stability. So how long this is going to be stable? After reconstitution, 7 days. If the bottle lasts for 7 days or 10 days, that product should be stable for 10 days. The stability in the sense, whatever is the control strategy adopted for the product should have that the chemical stability and microbial stability during that 7 days or 8 days after reconstitution. This is in-use stability. Sometimes the product has to be reconstituted with a particular solvent, like for example, injectables. They give particular solvent like some buffer solution or water for injection or normal saline or ringer lactate. So all these, the effect of mixing your product with these solutions like buffers or ringer lactate or sodium chloride solution or water for injection. So how this is effective, how long this is going to be stable once mixed with the drug product. So this is in-use stability. So this in-use stability also plays a role because you get a lot of complaints at the time of administration of this. If the drug product gets precipitated, then it's not fit for use. Then you get a complaint. So you have to clearly specify what are the solvents, what are the buffers, what are the solutions which can be used with this product, before diluting. So that has to be determined beforehand. Then another important aspect of this is the bioequivalence. So chemical equivalence is one, then the bioequivalence. So there are different ways to establish bioequivalence, either pharmacodynamic studies or pharmacokinetic studies. I think you guys understand what is a pharmacodynamic study and what is a pharmacokinetic study. Then the analytes, whether you evaluate or estimate active ingredient or a metabolite. The study design, whether it's a crossover design or parallel design. And what are the bioanalytical methods, whether your methods are suitable to estimate those picogram level, nanogram level in the plasma. Then what is the 90% confidence interval? So this 90% confidence interval is a statistical treatment to the data you generate while conducting the bioequivalence study. So it should be generally accepted 90% CI is, it should be between 80 to 125%, 80% to 125%. Your CI of the two pharmacokinetic parameters like Cmax and AUC. AUC 0 to T or 0 to infinity. So these are the two parameters, the ratio of which your product and the comparison product should fall within the 90% CI. Ratio should fall as well as your confidence interval should be between 80 to 125. So what are the challenges for pharmaceutical equivalency? So these are the challenges we face. The stability of the product. So sometimes the company sells a tablet. By the time the patient opens the blister or the strip, you find a powder. That means its physical integrity is gone. So product, something is wrong during transportation, during manufacturing, during storage, that we cannot estimate. So this has to be previously determined how the product has to be stored so that it remains intact till it is consumed. Then impurities, as I mentioned earlier, the impurities is a big challenge in pharmaceutical dosage forms. The impurities should be comparable with your reference product. And some of the impurities, as I mentioned, are very cancerous causing, they're mutagenic, it affects your health. Then the residual solvents, the dissolution, the dissociation constant, the scoring. This is another important thing. Some of the tablets we see are scored. There's a line dissecting the product. So if somebody was directed to take half of the tablet, they break along the scoring line. So if half of the tablet should contain exactly half of the ingredient, as well as the dissolution should be exactly the same like. So otherwise it doesn't perform as it was designed. So the scoring evaluation is an important part. When you break the tablet, it should not lose more than 3% of the weight of the tablet. If it loses more than 3%, it could affect the assay of the split tablet. So the split tablet study is an important thing. I don't know how far we control this scoring, because some patients are written half the tablet to take it. So half the tablet if they take, if 50 milligram tablet, they should get 25 milligram or plus or minus whatever is the tolerance. And the content uniformity. Each unit should have the same content. It is estimated by assay or by weight variation. It depends on the amount present in the dosage form. So these are the challenges for bioequivalence. Physiological factors, the pharmaceutical factors, how we design the product, at what pH it's going to resolve. And the most important thing is about the special category products like these, the MDIs, DPIs, topical product, nasal deliveries, transdermal patches. So these are very, very critical in terms of establishing bioequivalence. And there are a lot of testing required in vitro as well as in vivo. So this is very challenging. Sometimes the transdermal patches like lidocaine patches, say lidocaine patch 5%. So how much of the drug is delivered? It's less than 3 or 4 or 5% drug is delivered. 95% remains in the patch. So that could be dangerous for somebody who is handling the patch. So how much drug should remain in the patch again is an important criteria in establishing the bioequivalence, the performance equivalence. And there are certain highly variable drug products like fenofibrate or progesterone, where intrasubject variability is more than 30%. So they are very, very challenging. Unless the product is formulated properly, this establishing bioequivalence is very difficult. And there are narrow therapeutic index drugs like levothyroxine, carbamazepine. So there are many, theophylline. So they are very narrow therapeutic index. I think you guys understand narrow therapeutic index. Like the safety margin between the effective dose and toxic dose is very less. So that exactly has to be that narrow band from toxic level to subtherapeutic level is very narrow band. So this should meet that. Cmax and AUC has to follow with that. Then establishing in vitro in vivo correlates, this correlation, IVIVC. This is an important aspect because every time you cannot conduct bioequivalence studies. So indirectly, you should have a surrogate method which can establish bioequivalence in vitro. So there are certain tools and techniques available to establish in vitro equivalence. So for every batch release, you should follow IVIVC. Not bioequivalence, every batch you cannot conduct. So in vitro equivalency methods, they're available. So those methods should be used to establish in vitro in vivo correlation. Then what does a regulator expect now? So after understanding all these, so as a regulator, what do you want? You want a therapeutic equivalence, isn't it? So therapeutic equivalence means you expect a product should perform in the same way as it was intended or in compared to some reference product. It has to be a therapeutic equivalence. Unless it is therapeutic equivalence, you cannot ask them to be substituted. And most of the time we hear In India, doctors should be asked to write a chemical name of the drug. So who ensures those chemical names of every company are equal? That's a big challenge for everybody, for the pharmaceutical manufacturers and the regulators. So what is therapeutic equivalence? So first of all, therapeutic equivalence, to understand, we should understand what is a pharmaceutical equivalent first. The pharmaceutical equivalent is like Q1, Q2, has the same qualitative, quantitative ingredients, same dosage form, same route of administration, and same strength. So this is a pharmaceutical equivalent. So there are exceptions, like, you can change the release mechanism for a controlled release, or you can change the flavor, you can change the color, you can change the preservative, or your expiration date could be different. These are certain exceptions for these pharmaceutical equivalents. Then we should not get confused with pharmaceutical alternatives. So pharmaceutical equivalent is having the same ingredient, same dosage form, same level, but these are alternatives, like a tablet and a capsule, or a salt of one molecule to the other salt. Tetracycline hydrochloride, doxycycline hyclate versus doxycycline hydrochloride. So these are pharmaceutical alternatives. They're not equivalents. They're alternatives. Sometimes we assume that pharmaceutical alternative is equal to a pharmaceutical equivalent. So therapeutic equivalent, coming back, the pharmaceutical equivalent plus should establish, sorry, should have same clinical effect and safety profile when administered to the patient as per the labeling conditions. So this is the pharmaceutical equivalent plus the bioequivalent. This is the bioequivalence. So these are there are 5 criteria to say that a product is therapeutic equivalent: safe and effective, a pharmaceutical equivalent, bioequivalent, adequately labeled, then manufactured with CGMP. So CGMPs is the basic underlying principle for this product to be manufactured. So the labeling part is an important part, but we ignore in India. So this labeling, under what conditions the product is approved, so what are the labeling directions for the patient, whether there is a medication guide, the prescriber's information, the product is a risk product or not. So there are a lot of information that goes with labeling internationally, but we still need to catch up on this. So pharmaceutical equivalent, when we say, so equivalent to what? This is again, the question comes to our mind, when the press, the public, the politicians, the government ask us to insist the doctors to write a generic name. Then generic to what? Is it right to insist prescribers to write generic names for medicines? Can we ensure that all generic products sold in India are performing the same way? When we cannot ensure, so I don't know what to say an answer for this question. Can we insist the prescribers? Are all generic medicines pharmaceutical equivalent? Maybe pharmaceutical equivalent, but they cannot be therapeutic equivalent. Can we say that? Unless we evaluate, unless we apply science, unless we use tools to determine that therapeutically equivalent, we cannot insist anybody to prescribe a generic name for medicines. That is my personal opinion. I could be wrong. So recap. So typical product life cycle. So we've gone through that. Then what is the current scenario? So where do we stand? So this is my current understanding. I could be wrong. I could be challenged. So all the first 8 steps, in most of the cases, I don't say 100%, is not followed. And you get directly an application for manufacture without any of these. We don't lock in the API, we don't lock in the product, we don't lock in the manufacturing process, we don't know the stability of the product, but we get an application for approval. So then we review it, we ask questions, we audit them, we then approve, then commercial manufacture, distribution, then who is monitoring the product? The complaint handling, the pharmacovigilance, adverse reporting. So I don't know. So I could be wrong. So this is what I understand the current scenario. So challenges for skipping some of these steps in the product. So what are the challenges we face when a product life cycle is not fully followed? you get a variable product. So product will not be uniform all the time. It could be different shape, different size, and different performance. And stability of the product is a question. You get adverse publicity, and you get a lot of complaints from the market, like product is ineffective, does not work, and product is contaminated. I see a lot of particles in the product. There is a growth in the product, liquid products, and changes in appearance of the product. So these are all the consequences of not following proper product lifecycle management. So what are the burden for not following these product lifecycle steps, especially for the regulators? Now in India, once we permit the product manufacturing, like granting the license, and the manufacturer responsibility is just there. And it's your responsibility to continuously monitor the product, take samples, seize the samples, freeze the samples, use Form 17, 17A, 18, 13, then 15, 16, investigation, court cases, is all burden on the current regulators. The court cases, then you get a lot of questions from the legislators, press, government, whatnot. Everybody questions you, because once you license the product, the responsibility is on you. So what is the social impact of skipping these product life cycle? So credibility of the product is gone. If somebody suspects, then the confidence on the product goes. Confidence on the regulators will be lowered. Your self-esteem goes. So there will be disturbance in the supply chain. So once you get compliant, so you try to stop the distribution and manufacture of product that disturbs the supply chain. So these are the societal impact. So what is the need of the hour? So how can we mitigate these challenges on the regulators? So we need to just inculcate a positive attitude. So how we are approving the product, we need to introspect whether are we applying the science behind the product development and product testing and equivalency establishment. Are we following any tools? Do we apply any statistical principles? So this is an introspection is required for us. So we need to have a continuous understanding of what's happening outside world. So we have to adopt the proven regulatory techniques, proven regulatory approaches. There are many tools publicly available. We can adopt them without any cost to that. So what needs is the attitude change. That is the most important thing required for us. So again, going back to the first slide, the patients and the prescribers have a lot of confidence on the drugs, assuming that the products are safe, effective. So this is, once you adopt yourself, change your approval process, you can assure the prescribers and public that the pharmaceutical equivalent drugs are therapeutic equivalent. This is the ultimate goal of all of us. That is, we are ensuring public the availability of a quality product, assuring the public that they can take it, closing their eyes, the product that is regulated, approved, sold in India, or therapeutic equivalent without any undocumented adverse events. So undocumented in the sense that is not disclosed on the label, because all chemicals have some effects. So the benefit risk analysis, if you see, if the benefits are more, the product gets approved. So, but we disclose what are the consequences, what are the adverse events reported during the clinical study or the bioequivalence study, that is disclosed on the label. So that is the bottom line. So once we follow all these regulatory principles, we can ensure anybody that the pharmaceutically equivalent products sold in India are therapeutic equivalent. So with this, I conclude, and thank you very much for your time. And if you have any questions, so I'll take up.
Thank you very much, sir. Indeed, it was a very great, insightful discussion on the entire product life cycle of the pharmaceutical product. And you have touched down each and every minute, but yet very important aspects in the development of the product and the need for proper regulation of the product developmental stage at different stages in ensuring that the pharmaceutical product is equivalent enough, not only in the pharmaceutical equivalence, but also in the therapeutic equivalence. We are so much grateful for your detailed presentation, sir, and I believe all the participants have thoroughly enjoyed the grateful insights from your today's talk. Thank you once again, sir, for such an elaborate presentation. And now we are opening the floor for the questions, and I would request all the participants to pose their questions in the Q&A tab. Alternatively, you can also use the chat tab also for posting your questions, but once again, Q&A tab would be more preferable to take up the questions specifically. And if any of the participants would like to have an interaction with Dr. Veeraju sir, you may please raise your hands and we would allow you to speak for 1 to 2 minutes, considering the time constraints. In the meantime, I also recognize the presence of senior regulators from CDSU on state, Dr. Arvind Kukriti and PBN Prasad sir and Rajshekhar Malli and Shalum K sir and many other senior regulators have also joined from the different states for this today's program, and it clearly indicates how interesting the program and how in-depth the talk of Dr. Veeraju sir that the participants are enjoying. And sir, I could see a few of the questions, and one is from Mr. Kaushik Desai ji. I think he's a good friend of yourself, and he has been contributing to the Adcock Academy activities, and we had a fortune of his talk in our platform. So Kaushik Desai ji's question is that he would like to hear your views on the digitalization of quality and how Indian regulators should equip themselves with these digitalized systems.
Yeah, Kaushik, nice to hear from you, man. Very nice and a very good friend of us. Long time in IPA, and a lot of platforms we met, and it's a wonderful, interesting topic, Kaushik. Yeah, digitalization is definitely streamline the review and approval process all over the world. Even the Latin American countries, the Middle Eastern countries, South Asian countries, they adopted digitalization of all the review approval program that greatly reduces the effect of our scarce resources and standardizes the process and make quick decisions on the approvability or non-approvability of the products. Yes, definitely. There are a lot of technology, is available and evolving, and in terms of the quality management, document management, LIMS or quality document management systems, and the ECTD publications and ECTD requirements. So all the discussion, yes, all over the world, they're following. That is the reason I mentioned, we need to change our attitude to look into these aspects so that it is less burdensome on all our regulatory officers. Yes, it's a wonderful question, Kaushik, and nice to hear from you.
Thank you so much, sir. And there is another question from Mr. Arun Sethi regarding the cosmetics. I mean, with respect to the cosmetics, how extent that the cosmetics need to comply with these designs on quality systems? Yeah,
again, so the design principles are the same. If there are medicated cosmetics, they become like they are regulated like a drug. So like some of the creams and ointments, like acne medications, do you call them as a cosmetic or a drug? So acne medications they are regulated like a drug. 100% you need to follow the same quality product life cycle management for certain of these medicated cosmetics. But other cosmetics, yes. The two important things in cosmetic approval is the misbranding. You have to be careful about misbranding. So that is the most important aspect that is neglected all over the world. I don't say here. So you see a lot of advertisement that it fairness creams. How far they are going to work? Nobody has evaluated any data. So that is one part. Other part is we have to see that the ingredients they use or generally recognized as safe. GRAS, that's called the acronym they use is GRAS molecules or GRAS chemicals. So you have to adopt this and see that if there is an active ingredient present, yes, that should be regulated differently. If there is no active ingredient present, simply like a moisturizer, lotion or a cream, yeah, it's like a cosmetic, and you see that they are manufactured with generally recognized as safe ingredients. That's only currently, that is the only aspect that I can talk of.
Thank you, sir, for the explanation. So it's very clear that medicated cosmetics need to be followed the same principles like the drugs in terms of the product development and design aspects. Another question from Muthuswami Iyer. Can you please touch base on the co-excipients? Nowadays, there are two excipients mixed as a single excipients. And what are the critical tests or control to apply in case of use in the FDFs?
Yeah. So these co-excipients or proprietary excipients, you can call, it's a mixture of 2 or 3 that they're called proprietary excipients. Yes, you need to understand. So what is the composition of that and what is the function of that and what is the physicochemical characters of that composition? You have to see, you have to declare the individual components on the label, no doubt at all. This is like, your flavor. Flavor contains a lot of ingredients. 6 or 7 ingredients are mixed together, makes into one flavor. The qualitative, quantitative composition, sometimes they disclose, sometimes they don't disclose. But you have to have a particular identification test and say like pH or specific gravity or characteristic smell, or you can identify some functional group, depending on the information shared by these proprietary excipient manufacturers. Whether it could be flavor, it could be color, or it could be an excipient. Yes. The mixture of excipients definitely affect the performance of the dosage form. It all depends on the, I mentioned QTTP, quality target product. What you are aiming for the product should be achievable using this particular mixture of proprietary excipient. So this proprietary excipient, the manufacturer has to evaluate and decide what are the critical quality attributes for these proprietary excipients should possess. Based on that, they can proceed further. Like some of the excipients could promote bacterial growth. So microbial control is a critical element. The other one is your bulk density. If it is used for solid dosage form, bulk density makes a lot of difference. And another important aspect is about the particle size distribution, particle size, the D10, D50, D90, and the distribution. So these are the critical aspects, just like any other, either single excipient or a proprietary mixture of excipients.
Thank you, sir. Another request from Ms. Veena, that how IV-IPC studies are carried out for BCS class 4 drugs.
Oh yeah, so that is very specific. You can reach me. I'll explain to you. So that will be, that's a very specific, and it requires a lot of understanding of the BCS4 criteria plus what are the techniques available. So that takes a lot of time for us. We can share my email and you can reach me. We'll discuss with that. Okay.
Definitely, sir. So we'll touch base with Ms. Veena and facilitate her to reach out to you for the specific question. And there is also another comment from Dr. Bhartish Jagshetty regarding the digitalization aspects in India and the implementation aspects on the regulatory system in the current scenario. Of course, I mean, there are some challenges that we could, foresee.
Yeah, that's exactly. It's definitely it's going to help the industry as well as the regulators, in making faster decisions. The digitalization is the need of the hour. How soon we adopt, it all depends on the manufacturers' support and cooperation. Then the regulators are ready to take up this. So I think we can work together in digitalizing. So all our application process, review process, approval process, that definitely is going to help.
There is a final question regarding the herbal formulations. that how can we perform the evaluation test for herbal formulations from Rajeshwari. Sir, would you like to take this question? Or?
Yeah, that's fine. Herbal formulations, are we regulating this in terms of like our under Section 3B? Do they fall under Section 3B? I'm not sure, because this is again Ayush Ministry comes into place here. So when Ayush Ministry comes into place, so anything, generally, if you ask me a general question, yes, the product has to be safe and effective. Whether you can estimate the active ingredient or not, leave aside. What about other parameters like performance parameters, like if it is a tablet, whether it has to be dissolved or not, whether it should be free from microbial contamination or not. These are the aspects we need to evaluate. Forget about the chemical composition. If there is a currently feasible techniques available, definitely we can insist on that. If it is not available, there are many molecules like, even allopathy molecules, I'll tell you, the mixture of many molecules used for ADHD. There are many I remember more than 10 molecules. Chemically, you cannot evaluate separately, but as a mixer, following GMP principles say that is added into the product, that is certified, that is approved. Even in US FDA regulation, I'm talking about. So there are different techniques available to ensure the quality of the product. Yes, the industry has to be honest in terms of the ingredient evaluation, then addition to the product, then monitoring the product, not chemically, but other aspects, so that it doesn't cause any harm to the consumer.
Well said, sir. Thank you so much. And I think with that, we come to the end of the Q&A. May I now request Sri Deshpande ji, the Chief Advisor of All India Drugs Control Officers Confederation, for the concluding remarks.
Thank you, Lakshmi. It's really a great pleasure to hear Dr. Veer Raju, especially because he is seeing the dream which we saw together of establishing training academy, and it's really a pleasure and privilege for us and for him also to make a presentation on the platform of All India Training Academy virtually to start with. Hopefully, when this COVID-19 goes up, we can have one physical session wherein Dr. Veer Raju can again make the presentation. Coming back to today's presentation, obviously it was very lucid and very effective and informative presentation. Explain everything in a very simple manner. This quality by design or a pharmaceutical quality system is not that simple as is made out, but he has explained it in a very simple manner. Started with the quality, what is quality and what is expected by perception or whatever, and then product cycle. Once the product cycle was explained, then each critical element of the product cycle was elaborated by him, properly explained how API characterization affects the quality, analytical instruments, how audit trail are critical, important, challenges in dosage form, stability, storage, shelf life, bioequivalence. That's the most critical issue in India, at least, and he has rightly said because he was a regulator, so he has blended his experience as a regulator and now 20 years experience in the pharmaceutical industry to give a factual side, and he has presented it both sides, especially on bioequivalence, that there are large number of products claiming to be generic, or branded generics, and how we can ensure that they are equivalent to what? Equivalent to innovators' product or equivalent to each other. What is that? Today we only use the word generic in a very loose way, but not yet established. And challenges in equivalency, he has explained. Therapeutic equivalence, The in-use stability is another critical factor, perhaps not attended to. All in all, excellent presentation, most useful, and I'm sure that a lot of value addition to the pharmaceutical industry professional as well as regulators. To me, I think the today's sessions, what is the learning of today's session? At focus on product cycle, and if that is properly, as Mr. Dr. Veeraraju has said, that in India perhaps so many steps are not done and directly application for approval is made. So if as a regulator, if we start focusing on uh, proper product cycle before a product is approved, before it is brought in the market, a lot of hassles of drawing samples, issuing notices, sending counterparts, filing prosecutions will reduce. In India, it cannot be brought to zero, but it will be certainly reduced. The second learning is perhaps we have more than 5 lakhs distribution outlet in India, and we spend a lot of time in that, and we spend a lot of time, comparatively less time on manufacturing monitoring. The focus should be what is the status of quality by design in pharmaceutical industry. And we say that there are 10,000 or 5,000 or whatever, but we can, as a learning today, we can focus more, adopt formula 80-20, focus our 80% resources on 100 top companies, because they are occupying the 80% share of the market in terms of volume, in terms of price. So focus on those 100 companies, and the hassles of the regulators will be reduced. The confidence of the public in regulators as well as the product available in market will increase, and it's a win-win situation for pharmaceutical industry as well as the regulators. Thank you so much, Dr. Veeraraju, and hope to hear you again and hope to meet you in person. Thank you.
Yeah, thank you, Deshpande. Nice summarized you nicely, and it's wonderful, seeing you again. Thank you very much, and thanks all audience, for their time, spending on Sunday. So thank you very much, and hope to see you again. Bye.
Thank you so much, sir, and thank you so much.
Thank you, Veeraraj Garu. Thank you.
Thank you, Bhaskar. Bye.
Thank you, everyone, and I hope we have enjoyed the session thoroughly. And there are a lot of learnings, not only to the industry, but also to the regulators with this detailed elusive presentation and the key takeaways for the regulators, especially on the points illustrated by Deshpande. So with that, I thanks once again for all the participants for patient listening, and stay tuned to our further programs, and keep tracking our website for the updates on the presentation and the video recording of the sessions for those who have missed to attend the live program. Thank you once again. Have a good day.
Bye.
Bye-bye. Bye, everybody.
Bye, sir.
How long